Okay, good morning everyone. For today's lecture, this is all about nucleus. I am Randel. I'm going to be your lecturer for today's lecture. I believe that you're done already with the cytoplasm, no? So, tapos na kamo sa sabaw, subong kadto na ta sa unod. Okay? For our learning objectives, at the end of this lecture, it is expected that you all know the structure and functions of the nucleus. You need to be able to know the different phases of the cell cycle, compare the features of mitosis and meiosis, and know apoptosis and its features. And we will cover everything, no? The structure and functions of the nucleus, the cell cycle, mitosis, stem cells and tissue renewal, meiosis and apoptosis. Okay. So, kadto na ta sa nucleus. So the nucleus, remember that the nucleus is the most noticeable structure of the cell, okay? In a eukaryotic cell, remember? And it is the most defining feature of eukaryotic cells. So kabalo na kamo kung anong pinagkaiba ng eukaryotic cells and prokaryotic cells. Okay, so the nucleus, this green one here, is usually ito yung command center of the cell. Bakit? Because it directs everything. Okay? Siya nagda-direct ng paano mag-produce or kailan magpo-produce ng mga substances needed para in order for cell survival and cell division or cell growth. No? It contains the cellular genetic material. Okay? Kasi nandoon yung ano niya. Kaya command center siya kasi nandoon lahat ng mga genetic material needed for cellular survival and cellular growth. Okay, for the structure of the nucleus, remember that generally speaking, the nucleus is a large rounded or oval structure. However, may mga cases na hindi ito yung case, okay? And they are usually 5 to 10 micrometers in diameter. Okay? Ito yung, generally speaking lang ha. For example, in a squamous cell, yung squamous cell, 5 to 10 micrometers in diameter. So, just around the size of your red cell, which is 7 micrometers. So, isipin na lang na yung red cell, parang ganun yung size niya ng most of your somatic cells, okay? Most of the cells of the body, such as for example, yung squamous cells. And they can be uninuclear or multinuclear. Pag sinabing multinuclear, usually sa mga skeletal muscle ito when they form syncytium, no? So nagiging multinucleated yung appearance niya. So this is an example of your hepatocyte. Okay, this is a liver tissue, no? Meron siyang hepatocyte. So each one of these hepatocytes, meron siyang itong oval structure, which are your nucleus. Remember, yung cytoplasmic membrane, distinct and well-defined. And the nucleus, the oval nucleus, meron siyang mga darker areas and mga lighter areas, which we will later discuss kung ano yung mga yun. Okay? As I have said kanina, so generally speaking, these are large, rounded oval structure. However, there are cells in the body na hindi oval or hindi round yung structure ng nucleus, such as this one, okay? Yung mga neutrophils mo, these are your neutrophils, no? Remember na hindi rounded, but rather indented or madaming lobations ang imo nga mga neutrophils. However, during their younger days or younger stages, medyo round siya, nagiging round, indented, and eventually nagiging may mga filaments na lang connecting these lobations. Also, your red blood cell, wala siyang nucleus, no? Okay, pero during the younger areas or the younger stages of the red cells, meron silang nucleus na which are round or ovoid, then eventually extruded from the cell to become the mature erythrocyte. So, generally speaking, ito yung mga features ng ating cells, okay? Okay, so the components of nucleus, konti lang compared to your cytoplasm, which has many organelles, no? Medyo complex yung components ng cytoplasm mo. Pero with regards to nucleus, konti lang. So it contains nuclear envelope containing the chromatin, and a dense body which is your nucleolus, and then the substance that holds together, the structure that holds them together, the nucleoplasm. We will going to discuss each one by one. Okay, so we're going now to discuss the nuclear envelope. Okay, so the nuclear envelope, this one, itong green na na-highlight ng ating picture, that is the nuclear envelope. Remember that the nuclear envelope is selectively permeable. When we say selectively permeable, it only allows a certain macromolecule or a certain ion na pwedeng pumasok in and out of the cell. So parang kayo, no, mga mapili ba, mga pida kung sino yung juwain, okay? Ito dapat ganito, dapat alam, dapat hindi alampuan, ganun ba? So selectively permeable just like the cell. So innate talaga sa atin na selectively permeable or pilian, okay? And remember, the nuclear envelope is a double membrane. Okay, remember there is also another organelle that is known to have two membranes or double membrane, which is itong parang capsule na to, or itong parang structure na yan. This is your mitochondrion, okay? So just like the mitochondria, the nuclear envelope is a double membrane. So it has an outer layer or outer membrane. Okay, itong outer layer na to, which is usually closely associated with your rough endoplasmic reticulum. Itong rough endoplasmic reticulum, it is said to be rough because of the ribosome studded sa surface niya. Okay, kaya siya rough endoplasmic reticulum. It is continuous with your outer nuclear membrane or nuclear envelope. And then sa gitna niyan is the perinuclear space. Later, I'm going to show you another close-up picture of that. And the inner membrane of your nuclear envelope, okay, itong sa loob, okay, closely associated with your nuclear lamina, which is parang net siya, no? Composed of your proteins named as lamins, okay? And we also have nuclear pore complexes. Okay? Ito yung nuclear pore complexes, yung pinaka-main protein dyan, called nucleoporins. Ito yung nuclear pore complexes na yan. It traverses through the nuclear envelope. So it is an integral protein. Remember that proteins natin are two types. No, we have the peripheral and the integral protein. Usually peripheral can either be found out superficially sa labas or sa loob. Pero yung integral protein, it traverses through the nuclear envelope. So this is an example of your integral protein. And the major protein contained or composing the nuclear pore complexes are your nucleoporins. Okay, remember. And also yung movement ng mga macromolecules and ions, these are bidirectional by way of your or through your nuclear pore complexes. Okay? And again, yung nucleoplasm which holds these components together. Okay, so as I've said, ito na yung clearer or zoom na mga pictures natin, okay? Again, so the nuclear envelope is a double membrane, right? So it is composed of an outer membrane, outer layer, and an inner layer, and within that is the perinuclear space. Okay, remember, it is closely associated with your rough endoplasmic reticulum, while your inner membrane is closely associated with the nuclear lamina composed of your proteins termed as lamins. Okay, and this is the nuclear pore complex, and the major proteins composing it are your nucleoporins. Okay, and then enclosing it are your chromatins and the nucleoplasm which holds them together. So this is a transmission electron micrograph. Remember, ito yung NPC is the nuclear pore complexes. Remember this one? Yan. Yung mga nuclear pore complexes yan. And these net-like structures are your nuclear lamina. Okay? So, chromatin. Ano naman yung chromatin? Chromatin is a complex of DNA and proteins. Remember, we're going to discuss or we will later take up ano yung mga proteins na yun associated with DNA. And the chromatin forms the chromosomes within the nucleus. And remember, meron tayong dalawang forms of chromatin. These are based on the transcription activity nila, no? So there are two forms of chromatin. We have the heterochromatin and the euchromatin. Later we will going to discuss about it. So just to show you the structure of the chromatin, so we start off with the double helix DNA. So this is the double helix DNA, 1, 2, and then yung mga bases natin, okay, mga base pairs. Okay, sila yung parang sa ladder ba, okay? And then they wound up around this structure here. Pero pag tinanggal mo ang panit, yun, you will be able to see these round structures in this drawing. These are your histones. These are the proteins associated with your DNA. And then itong grupo ng 8 na histones, kasama ang napalibot nga DNA is called nucleosome. Okay? Nucleosome. Ito yung subunit ng DNA and proteins, nucleosome. And again, yun, itong isang strip ng histone or ng nucleosomes, it is called the chromatin, okay? Or chromatin fiber. And then, sila ay magiging either loosely packed or densely packed based on the transcriptional activity of the chromatin. Okay? And then they will form the chromosome. Okay? So, as I have said, so there are two forms of chromatin. We have heterochromatin and the euchromatin. Remember that the heterochromatin, okay, tandaan nyo lang, HD, heterochromatin, these are densely packed. So, high density, okay? Heterochromatin, these are densely packed. So, ibig sabihin, busok sila. Kung sa puto pa, busok ba, hindi siya airy, okay? Densely packed. So, since densely packed sila, when you look at it under the electron microscope, or when you stain it using H&E, usually they stain darkly, okay? Kasi, yung mga DNA, mga acidic man yan, so basophilic yan, they will attract they will attract the basic stain, the hematoxylin, right? Okay, so densely packed sila, nakatago sila, kaya sila close. So you are curled up lang sila, okay? Just like sa sa tao, 'di ba? Inactive 'yon pag mga ganon, no? Ibig sabihin naka-curl up lang sila sa bed nila, yung tao na medyo hindi ba active, naka-curl up lang densely. So naka-curl up ka lang sa kwarto mo or sa higdaan mo, hasta dun ka na lang, yung eye bag mo nag-stain darkly na. So, yun yung mga inactive sa life. So, just like sa heterochromatin, these are transcriptionally inactive and hence they are called closed chromatin. Okay? Remember also that a form of heterochromatin present only in females are your bar bodies. Okay? Yung remember that the female sex chromosome, dalawa yun, di ba? Dalawang X chromosome. Okay? X chromosome. Ibig sabihin, yung isa dyan is an inactive form of your chromatin, which is your heterochromatin. So, sometimes ginagamit itong bar bodies to check if the individual, for example, in forensics, no, may makuha tayong mga sample, mga DNA samples dyan, or mga cells pa dyan na viable pa, okay, makikita natin na i-check yon kung female nga ba talaga, you check for the bar bodies. Usually, these are darkly staining around the nuclear membrane or sa periphery na nucleus. Okay, for example, then ano pa ba yung pwedeng i-check natin if female yung isa ka tao. For example, may manananggal, okay? Syempre manananggal putol, hindi makita kung anong meron sa iya, kung is it a female or a male genitalia. Wala lang. Gusto mo lang malaman kung isang manananggal ay babae or lalaki. Although pag sinabing manananggal, babae na. Pag lalaki siguro manananggol, okay? Anyway, yun gagawin mo is you stain, you smear pala yung buccal cavity niya, you stain it, okay? Pagtingin mo, pag may nakita kang stained or darkly stained structures around the nucleus, okay, yun, bar body, inactive X chromosome to. This must be a female manananggal. Okay? So, so pag may nahanap kayong manananggal, i-check nyo yun kung female ba to or hindi by identifying the bar body. So, this is an example of your nucleus, no? So, this is an electron micrograph again. So, as you can see, meron tayong this clump of darkly staining structures here is your nucleolus, okay? And again, there are still darker areas, no? Itong mga darker areas na to, these are your inactive chromatin or your heterochromatin, okay? While the surrounding lighter areas are the loosely packed euchromatin. So, again, same din, in contrast to your heterochromatin, parang tao lang din, loosely packed ka, okay? Hindi ka nagabulubid-bulubid ba? Tama ba? Bulubid-bulubid, loosely packed ka, so you are open. So, kung open ka, pwede kang magmingle o pwede kang maging active, transcriptionally active. So, pasok kayo, I'm open, no? So, euchromatin is open. Okay, so this one is an example of your bar body. So remember, this darker stain, this is a squamous cell, okay? Nakita mo, meron siyang darker areas around the nucleus, at the periphery of the nucleus. So that is a bar body. So this cell actually came from one of the top 10. It came from a female, okay? So you must know that. And then, ito yung histones natin. I have shown you this picture, the same picture actually, but I want to show you, nagko-coil sila. Laging coil yan. From the start, nag-helix, okay, coiling around the histones, and then eventually magko-coil ulit, and then mag-supercoil ulit. Yung supercoiling na yan, it will eventually form your chromosome. Okay, just to show you again the picture, ito yung ano natin, no? From DNA, the nucleosomes, the unit of DNA and core histones, and then they can be packed to form your chromatin fiber. So ito yung mga loops natin. And then these loops can actually be loosely packed. So just like this one, no? So loosely, and then yung heterochromatin na bulibod-bulibod ba. So, kaya densely packed siya. So, from this drawing alone, makikita mo, they are densely packed and they, therefore, should stain darker using H&E. In contrast to your euchromatin na wala siyang hindi siya densely packed. Okay? Loose siya. Okay? So, this the structure of your histone. So, usually, core of 8 histone molecules yan. Tandaan nyo na. 1, 2, 3, 4, 8 yan. And then there is a linker histone, the H1, which links na doon nakalink ang iyong DNA. Okay, yung double helix mo. Okay, remember the structure, 8 histone molecules. And this one, okay, this is a chromosome before replication, and you have the centromere, the central, actually hindi naman central kasi some of the chromosomes can be acrocentric, subcentric, no? Not necessarily central siya, okay? So, after replication, magpo-produce siya ng 2 chromatids. And remember, meron tayong short arm and long arm. Ang short arm also termed as the P. Bakit P? Tinawag siyang P kay putot. Okay? Putot for P. Or, English sang putot, petit. Okay? So, kaya P arm. Tapos, long arm is Q. Wala na silang may pangalan. No? Ipangalan taman sa sige, sunod na lang ng P, Q. O de, Q arm is long arm. Okay? Now, we go to your diploid and haploid. When we talk about the chromosomes, nakikita natin usually, ano ba yung diploid versus haploid? So, this is a simple table which shows you the characteristics of your diploid and haploid number with regards to the chromosome, no? Usually, pag diploid, 2N yan, wherein N is the number of chromosomes. Pag sinabing diploid, di, or 2 sets of chromosomes yan. So, that means 2 times 23 chromosomes, 46 chromosomes sa isa ka-human being. So, this is the number of chromosomes in a human. So, usually, pag diploid, somatic cells yon. Okay? And, sa diploid yun, 44 autosomes and 2 sex chromosomes. It's either XY or XX. Pag haploid naman, syempre hap, from the word hap, na half siya only and one set of chromosomes. So that means 23 chromosomes, 22 autosomes, and one sex chromosome, either X or Y. So usually yung haploid cells natin, these are your gametes. Okay, this can be either sperm or egg cell. One set of chromosome lang because during meiosis, okay, pag nagproduce sila ng offspring, magko-combine sila. Okay, from 23, 20 plus 23, magiging 2 sets ulit ng chromosome. Okay, remember that one ha. Exam magnet yon. And now we go to karyotyping. So ano ba yung karyotyping? Usually yung karyotyping, these are done actually ginagawa yun pag i-arrest yung cell cycle or cell division, usually sa metaphase yun, by using colchicine or anything that would stop the cell division by ways, kunwari sa microtubules, i-attack yung microtubules para mag-stop yung movement ng mga chromosomes natin. So just like that, sa colchicine, so na-stop, na-arrest yung cell cycle. So in the stages usually of metaphase, no? So anong ginagawa is that pinoposition yung mga chromosomes. Once na na-arrest na yung cell cycle, in-snip off yung mga chromosomes natin from the larger, from chromosome 1 up to the smallest, no? And then yung pinaka-last is the XY. Or the sex chromosome. So usually dito natin makikita yung mga chromosomal abnormalities. Usually kung kulang ka ng chromosome, for example, wala kang chromosome 13. So makikita mo yan, blanco yan. So meron kang either Batow syndrome, kung wala kang chromosome 8 or ano pa diyang mga chromosomes na kulang. For example, sa sex chromosome naman, nakita mo na map out mo na yung mga chromosomes natin, wala ka na diri sang Y chromosome, wala din X chromosome, so nagiging XO, so Turner syndrome. So makikita mo dyan yung karyotyping. Very important ito for mga chromosomal abnormalities. Okay? So this is the karyogram. So ibig sabihin, this one is genotypically male ito kasi XY. Okay? Bakit genotypically male? Kasi nakikita mo yung genes niya or nakikita mo yung lahat ng structures ng chromosomes niya, di ba? Kasi that can be, yung genotypically male, pwede kasing iba yung phenotypically male, no? Kasi, for example, na-raise up siya as a male nung bata pa siya or nung baby pa siya, pero yung ano pala niya, yung genotypic composition niya is female pala. So, yun yung may mga aberrations usually sa sex chromosome. Okay? So this one is a genotypic male. Okay? So 23 sets of chromosomes or 23 chromosomes, okay? 22 autosomes and one set or one pair of sex chromosomes. So pag binilang mo yan, 46 lahat yan. Okay? Pag may sobra na, magduda ka na, may problema sa chromosomes mo. Okay? Okay, now we're done with nucleus. We go now to the nucleolus. The nucleolus here is the green structure here. Okay, so these are spherical and highly basophilic. Again, basophilic siya kasi acidic, no? RNA will attract the basic stain or hematoxylin. Kaya basophilic siya. And it is active in protein synthesis. Remember this one, ha? Ribosomal RNA production is a responsibility of your nucleolus. Now we go now to the functions of the nucleus. So again, the nucleus controls the genetic information. Usually during meiosis ito, di ba? Kasi this will determine kung ano yung mga traits na pwedeng makuha sa mother or sa father. So meron tayong exchange of genetic information during the crossover in one of the phases of your meiosis. Okay? And then control of protein and enzyme synthesis. Again, since the nucleus is the command center, so it controls everything, including the cell division and cell growth. Siyempre, kailangan mo ng proteins and enzymes para mag-divide or mag-grow. Okay? And then storage of DNA, RNA, and ribosomes. Regulation, transcription of the mRNA to protein. Okay, transcription of mRNA to protein, and then production of ribosomes. Again, the function of your nucleolus yun, as I have said earlier. Okay? Now we go now Now we go now to. O dalawang now na. Kaya now na talaga yan. The cell cycle. So remember that each cell, meron siyang pagdadaanan. Gaya sa atin, may pinagdadaanan sa buhay. So parang human lang din, no? From starting ka sa fetus, for example, sa zygote, naging embryo, ganyan. Tapos naging baby, you naging child, adult, middle age, and then ano yan, matanda, tapos nag-reproduce ka, then mamamatay yung matanda, then again, cell cycle. So just like the human being, no? Cell cycle is a sequence of events that controls cell growth and division. Remember, pag sinabi mong sequence, isa-isa yon, step 1 to step 2, okay? So hindi pwedeng mag-skip, and it usually has 4 distinct phases from G1, S, G2, M. There is one phase which is G0. Nandoon nag-stay yung mga quiescent cells. So quiescent stage ito na kung saan nag-differentiate na yung mga cells natin. Pag sinabi mong differentiate, ibig sabihin napunta na niya yung gusto niyang maging na cell. For example, naging na siya, cardiac cell na siya. Okay? Naging cardiac myocyte na siya, naging brain cell na siya, okay? So ito yung, just to show you the drawing, no? G1 is the longest phase. As you can see it here. Later, we're going to discuss anong nangyayari during this phase. And then, nuclear or DNA replication in the S, synthesis phase. Then G2, and then back to mitosis. Okay, remember, ang shortest natin, itong G2. Okay? And then interphase, before ng mitosis is the interphase, the longest siya with regards to the mitosis, ano? And then some of the cells actually after mitosis will undergo quiescence, unless pag may cell injury, papasok sila ulit back into the cell cycle. Pag wala, they remain in the quiescent, parang dormant lang, waiting, Or sometimes nagiging pumupunta sila sa G0 when they become differentiated cells. Okay? Just another drawing or another cartoon showing you the cell cycle. Okay, remember G1, S2 duplicated, okay, G2, and then mitosis, itong phases natin later we will discuss, and then cytokinesis. Okay, cytokinesis is the cleavage, no? Hindi lang boobs ang may cleavage, pati yung cell, yung daughter cells natin, which will eventually separate into 2. Okay, so ito yung summary natin ng phases of the cell cycle. So G1 again, this is the longest and most variable phase. Ano ba nangyayari sa G1? Usually sa G1, syempre in preparation for the S phase, for the DNA synthesis, kailangan ng cell na mag-accumulate ng mga enzymes and nucleotides required for DNA replication. Okay, doon nagpo-produce ng mga materials needed for synthesis, which will eventually, pag okay na, it will proceed to the S phase, which is your DNA synthesis, okay, which includes DNA replication, histone synthesis, and beginning of centrosome duplication, no? Okay, replication, no? So, duplication, kasama na din yung centrosome doon. And then, pag natapos na-duplicate na yung DNA or nag-replicate na yung DNA, ngayon, magpo-proceed na naman sa G2 or growth phase. Ano na naman yung meron sa G2? Again, mag-accumulate ulit tayo ng proteins required for mitosis kasi kailangan mag-prepare tayo sa actual na talaga na cell division kasi ibibigay mo yun sa isang pang cell kasi mag-divide yun ng dalawa. So, you must have more proteins para ma-distribute sa dalawang daughter cells, right? Okay, now we go to mitosis. Ito yung actual cell division. And again, another phase, the quiescent phase or the G0 in which there is cell cycle arrest when the cells are terminal differentiated or in quiescence. Unless pag may injury, hindi sila papasok back to the cell cycle, back to G1. Okay? Now, cell cycle checkpoints. Remember that meron tayong mga cell cycle checkpoints kasi we need to check parang kumbaga may quality control sa lahat ng bagay sa steps na ito. Just like sa atin, di ba? Pag may mga manufacturing company, for example, sa cabinet maker, kailangan may mga checkpoints like this one. For the starting checkpoint, G1S checkpoint. So, tatanungin niya ng katawan or ng cell, okay ba yung nutrition natin, yung size, environment, favorable, pwede ba tayong makapag-proceed sa DNA synthesis? Okay, DNA, ang intact ba yung DNA natin before natin i-replicate ito? Okay, just like sa cabinet, o okay ba yung mga plywood natin, mga raw materials natin, mga lansang, mga martilyo, okay. Okay po. O sige. So, proceed tayo sa synthesis, DNA synthesis. Okay, mag-ano tayo? Sige, mag-produce na tayo ng mga cabinet. Okay. And then, after dun ma-produce na, ma-duplicate na yung DNA, meron again, G2M checkpoint. So, before it proceeds to mitosis na, okay, check natin kung kompleto na ba yung DNA, kompleto ba yung mga lansang na nalansang sa cabinet natin. Okay ba yun? Okay. Pag okay, sige, proceed tayo sa mitosis. Ngayon, what if hindi okay? Okay, what if there are may mga problema sa DNA replication, may mga bases na nagkapalit-palit, so iba na yung proteins na na-produce, okay? So nung gagawin, there are actually repair mechanisms yung DNA. Wais din yung cell natin, no? Meron siyang repair mechanism. So it's either i-snip niya, may mga endonucleases, i-snip niya yung portion of the DNA that is defective. So may mga mismatch repair genes na tayo or mismatch repair mechanism sa DNA replication. Okay? And then eventually, magpapasok na siya sa mitosis. Ngayon, pag sa mitosis, okay ba? Intact pa yung DNA? Naka-attach ba tanan sa mitotic spindle? Okay? Kasi pag nagka-problema, for example, merong gene na yung ilong mo gali na adto na sa buli. Anong himuon sang cell? So, tanggalon niya yung part na yan. I-tsakto niya sa position or i-snip out niya yan from the chromosome so that the cell can produce the cell can produce another cell na tama, normal cell. So ito yung mga problems natin with regards to cancer, okay? Pag sa cancer kasi, proliferative activity ng mga cancer cells natin ay sobra-sobra because wala nang checkpoints. Naging abnormal na yung mga checkpoints. Hindi na siya nagche-check na okay na ba itong, yun pala, yung mga lansang pala, mga tiko, yung mga lansang sa cabinet. So, wala naman nagche-check. So, po produce mga cabinet na may mga lansang. Okay, mga lansang na tiko. So, hindi okay, abnormal yung mga cabinets natin. Just like that, parang same lang din yung sa cancer cells. That's exactly actually na nangyayari sa malignancy, sa mga cancers natin. Okay? So, we go now to mitosis. Okay, mitosis is the actual cell division. And remember, ito yung the only cycle that can be observed in light microscopy. Remember, some of the cancer cells, actually, madami mga cancer cells na kinakount natin yung mitosis. For example, the number of mitosis per 10 high power fields. Sine-check natin yan whether this cancer is highly proliferative. Pag highly proliferative, ibig sabihin we can parang masabi natin na this is aggressive. Okay? So yun, mitosis nakikita natin doon. And usually remember that the parent cell divides and each of the two daughter cells receives a chromosomal set identical to that of parent cell. Remember, naka-highlight itong or naka-bold itong identical. Kasi pag sa mitosis, usually mga somatic cells, kung ano yung itsura sang parent cell, yun din dapat yung sa daughter cell. Like, for example, sa skin. So, squamous cell, ipoproduce mo, magde-divide yung cell, squamous cell pa din. Okay? So, identical yun. Yun yung mitosis natin. Okay? Again, this is just a structure of your chromosome. We have the putot, P arm, short arm, and the Q, long arm. So, primary constriction, the centrosome, and the secondary constriction here. And then sa tip niyan, these are your telomeres. Okay? Okay, ito yung sa cultured cell natin. Okay? So you can see it here. This is prophase. Ang ganda, no? This is cultured cell, no? Yan, sa prophase, nagko-condense na yung mga DNA natin. And then, pag sa metaphase, usually they align. So, tandaan nyo yung mga terms na yan. Nag-align sila at the center. And again, yung anaphase na yung centrosome or centromere natin, nandun sa pole, okay? Sa both poles. And then telophase, eventually, magdi-divide na siya, okay? Nakita mo na nandun na siya, oh. Okay? And then eventually, ended up by cytokinesis. Okay, so these are the summary of your mitosis phases mo. Tandaan no, pag sa prophase, before pro, no, the chromosomes condense, so kailangan natin i-disassemble muna yung nuclear envelope kasi ang mangyayari is lalarga yung mga yung chromosomes natin. So saan sila lalarga? Sa meta. Sa meta, mag-a-align sila sa middle, no? M for middle. So they will align at the center, at the middle of the cell. And then si anaphase naman, pupunta siya to separate poles. So si Ana, pole dancer yan, okay? Nandun, hindi lang siya gusto isa ka pole, duwagid ka pole ang iya nga sa utan. So on one side and the other side. So two poles, nandoon yung centrosomes, okay? Remember the organelles responsible for this, yung mga centrioles, microtubules natin, ha? And then lastly, yung telophase in which the nuclear envelope reforms around separated chromosomes and ends with cytokinesis. Again, as I have said, hindi lang boobs yung may cleavage, pati yung daughter cells. My God, wag nyo akong isumbong kay kay Miss Jem, baka kung anong sabihin, anong ginapang tudlo ko di sa inyo. Okay, sige. Hindi, I just want you to, para may association kayo, is better for your, for recall. Okay? Now we go to stem cells. Ano ba yung stem cells? Remember, hindi ito yung cells ng stem, ha? Okay? Stem cells, these are pluripotent. Okay? These are primitive cells that have the potential to differentiate into different cell types. Again, as I have said, pag sinabi mong differentiation, ang cell ay magiging committed to become a specific cell type. For example, magiging blood cell siya, magiging red blood cell siya, yung isa magiging neuron, yung isa magiging cardiac cell, okay? Isa magiging columnar cell, for example. And the stem cells divide in asymmetric manner. Pag sinabi mong asymmetric manner, ito yon. So magde-divide siya ng two cells, yung isang cell will be back into the stem cell pool for further or for future regeneration. Yung isang cell naman, ito yung magiging committed. Okay? Magiging committed siya to become a different cell type. Kaya tinawag siyang asymmetric. Pag sinabi kasing symmetric, once na nag-divide yung isang cell, yung two daughter cells, again, will divide into another cells. Ganon. Wala siyang pool na mababalik. So this is important yung stem cells kasi so that yung body natin may reserve na stem cells para kung sakaling may injury, we could be able to regenerate, we could be able to produce another cells for differentiation. Hindi tayo mauubusan, okay? So, ito yung pictures natin, okay? So, stem cell natin will divide. Again, may arrow tayo pabalik because this arm here will become again will become the reserve or mababalik siya sa stem cell pool for further division para maging another cell types para may pool tayo in cases may injury. And then the one side here will become the committed progenitor cells, okay, that will become differentiated cells. So this one columnar ciliated, okay? Remember also that stem cell, stem cell therapy, 'di ba? Meron tayong mga placenta, mga placental soap, mga ganun. Placenta soap pala, ganun. Actually, yung stem cell therapy, very proven na din yan actually kasi meron mga researches na diyan, very promising itong technology na to, itong innovation na to, in which yung cell ba, pwede siyang yung stem cells taken from yung mga primitive cells na taken usually yung nga sa mga fetus, ganun ba. So yan talaga. So pwede silang maging other cell types such as may mga rheumatic heart disease. Okay, may mga rheumatic heart disease, pwede yung stem cell maging committed to become valves, okay, to become the heart valve cells or the cells that make up your heart valves, yung mga fibroblasts natin, pwedeng yun. Okay? I believe meron na talaga yan na stem cell therapy. Okay? So, ito yung another example, no? Pluripotent stem cells. As you can see, merong arrow back because pag nag-divide siya, merong asymmetric cell division. One of the arm in the division of the stem cell will become the pool, okay, of your stem cells for future regeneration. And then the rest will become, or the other side of the division can become committed to form other cells of the body. So, pwedeng muscle cells, nerve cells, etc., etc. Okay? So, very clear on that. And now, with regards to tissue repair and tissue renewal, the different types of tissue can be divided accordingly. Okay? There are 3 types. So, aralin nyo to. When we say labile cells, di ba, parang sensitive siya, labile, okay? These are cells or tissues that are continuously dividing. So, ito yung normally ba, mabilis yung turnover ng cells, okay? Such as your hematopoietic cells, yung mga blood cells natin, and the surface epithelia, the skin and uterine lining. Remember, every day, di ba, pag panglugod ka, may mga buling ka. So, yun yung turnover ng cells. Mabilis mag-divide yung skin natin. Mabilis mag-divide yung epithelia natin. Kaya, nasa slough off agad yung mga cells natin. These cells actually can regenerate after injury as long as the pool of stem cells is preserved. Okay? Remember also na itong mga continuously dividing cells, ito yung mga targets ng natin, ng mga chemotherapy, no? Kasi yung chemotherapy, yung systemic chemotherapy will target yung mga malignant cells that are highly proliferating. Although kasi yung cancer therapy natin, hindi man siya talaga na specific, no? Ibig sabihin, it will target lahat ng mga highly proliferative cells, including the normal cells, which are your bone marrow, or your hematopoietic cells, and your skin, and your surface epithelia, yung skin, yung hair mo. Kaya, usually mga cancer patients, prone to infection, kasi affected na yung bone marrow nila, yung mga cells to fight the infection, medyo decreased na, and then nakakalbo sila. Kasi, affected yung mga continuously dividing. However, meron mga bagong mga therapy for cancer, no, yung mga targeted therapy which will only target the cancer cells, hindi yung mga normal na mga continuously dividing. However, these are medyo mahal yun, okay? Pero yun, promising din yan sa area of medicine, no, yung mga targeted therapy. Ganun na usually yung mga mga high-tech or mga, tawag dito, mga, basta, hindi ko masabi, yung mga methods na usually na ginagamit sa buong mga advanced na methodology, okay? So, done with labile, we go to the stable. Yung stable naman, ito yung mga pumapasok sa G0, in the quiescent, in the dormant phase. Meron silang minimal activity, okay? Minimal proliferative activity in the normal state. Pwede silang pumasok sa G1 or pumasok sila sa cell cycle pag may injury. Remember, ito yung mga stable cells such as your stable tissues such as your liver, kidney, and pancreas. Remember that the liver can regenerate. Diba? Usually, diba, pag may mga liver transplantation, yung donor, pag tinanggalan ng isang lobe, let's say right lobe na lang natira, it will regenerate some degree. Okay? Depending on the demands of the body. So, the liver can regenerate actually, but it's still hindi ganun ka highly proliferative as your labile cells. And lastly, your permanent or thermally differentiated and non-proliferative cells. Okay? Remember, yung thermally differentiated, ito na yung last na stage niya. Ito na, brain cell na ako. Cardiac cell na ako, skeletal muscle na ako. So, hindi na ako magpa-proliferate. Remember na hindi talaga magpa-proliferate yung mga cells na yan. For example, yung brain. When there is injury to the brain, for example, na-operan, may tumor, and part of the hemisphere is removed. So, yuhob na yung ulo mo. Hindi na yan hindi na yan magre-regenerate. Okay? But, however, pwede yang ma-stimulate yung brain natin. Kasi usually, pag sa adult age, nag-a-atrophy yung brain. Pero you can still stimulate, pwede natin ma-stimulate yung mga brain cells natin by engaging in activities na yung mind, mind triggering ba? Such as, yung pag-aaral. Yung mga pag-aaral, mag-aral tuon, mga quizzes natin. Pwede din actually yung ML, 'di ba? Maglaro. Okay? Pero wag niyong patamaan sa laro. You study first, okay? And then, yung heart. So, hindi ka na talaga. Pag hindi na proliferative yung heart, pag nasira yung heart mo, hindi na yan pwede unless ita-transplant. Okay? Now, again, meiosis, I have uploaded meron akong video na in-upload doon. Maganda yung ano niya doon, maganda yung explanation ni Amoeba Sisters doon. So, meiosis, meron tayong 2 successive cell divisions divided into 1 and 2, and again producing gametes, which are your sperm and egg cells. Meiosis is termed as reduction division kasi nagiging haploid eventually from 2 sets of chromosomes, naging 1 set of chromosomes na lang, okay? And remember, in contrast to mitosis kanina, which is identical cell, yung example natin kanina, squamous cell, mag-produce siya, mag-mitosis, mag-produce siya ng mga daughter cells na squamous cell din. Dito, magkaiba, non-identical haploid cells because, as I have said, during the earliest phase or sa interphase before meiosis, mayroong mga synapses and crossing over na kung saan nandun yung genetic makeup na may exchanges in genetic information between the chromosomes. Okay? So these are your gametes, the ovum and the egg cell and the sperm cell. So this just shows you yung meiosis 1, eventually dalawang 2 sets of chromosome, and eventually isa na lang. Yung daughter cells natin, isa na lang. And remember, there are exchanges in the chromosomal information. Kaya nagiging kahit magkapatid kayo, iba yung itsura nyo. Yung isa inchik, yung isa bukaw. Okay? Unless siguro kung kayong manggi ka mo or itom ka mo or nigra ka mo or dark-skinned ba kayo, okay? Tapos biglang may blonde, tapos blue eyes, magtaka ka na, okay? Sige. Now, it is important to know the difference between mitosis and meiosis. Very short lang yon, okay? Again, just to emphasize, mitosis, diploid cell, an identical cell, as I have mentioned, yung squamous cell natin kanina na example, and they produce somatic cells such as yung mga skin cells natin, the cells lining the epithelium, the cells lining the gastric epithelium, the uterine lining, and remember, only 1 cell division. This is in contrast with your meiosis, which will produce half of the chromosomes, haploid cell, and non-identical because there is synapses and crossing over that occurs at the interphase before the meiosis, and 2 cell divisions producing gametes, egg cell, and sperm cell. So remember this, kahit pakayon pakulbon, kabalo ka mo, kahit nabunggo ang inyong ulo, pag tinanong, produce only 1 cell division, mitosis. Okay? So this is your picture again. Okay, now we go to apoptosis. So, yung apoptosis kasi, sometimes during development, for example, no, sa aton, during the fetus, syempre yung kamay, yung fingers natin, meron tayong mga webs pa dyan, okay? Sa bata, yung webs bala, yung webs sa finger natin, yung parang sapato, yung tiil sapato, may web siya, di ba? So, during our sa embryo natin, sa maliit pa tayo, no, may webs tayo dyan. Eventually, during apoptosis, natatanggal yung webs na yan. Okay? Yun yung apoptosis. Okay? Apoptosis is actually programmed cell death. So, ibig sabihin, itatag ng katawan na may problema itong cell na to. Okay? May something talaga sa cell na to na may basta abnormal itong cell na ito. Sige, itatag natin yan. That is programmed cell death, apoptosis. So again, redundant or defective cells are eliminated without local inflammatory reaction in the tissue. In contrast to necrosis, kasi pag necrosis, yun necrosis ba, namatay ang mga cells mo, surrounding cells. Hence, nag-i-incite siya ng inflammatory reaction. Secondary to injury, ito usually yung necrosis natin. Okay? Again, as I have said, yung webs natin, no, redundant cells. So, ibig sabihin, yung mga webs na yon, yung web tissue na yun, before tayo mag-develop talaga ng complete sets of yung hands natin, na complete set of fingers na wala ng webs, yun, nag-apoptosis yun. Unless siguro hindi pa nag-apoptosis yung mga kamot nyo, nga daw parihas pa din sa sa pato. O, tanawin nyo yung mga kamot kung may mga webs pa dyan. Okay? Again, so wala siyang inflammatory reaction, right? There are production of apoptotic bodies that are phagocytosed by your macrophages. Okay? And again, these are controlled by proteins under the BCL2 family. So ito yung mga example nito are BAX and BAC, BAX and BAK. Okay? So just to show you and contrast between necrosis and apoptosis, yan, so for example, this is the viable cell, and the cell shrinks in apoptosis, yung chromatin condensation, magsicellular blebbing, or membrane bleb siya, and then eventually magpa-fragment siya, and then magbabuds. At then itong mga budding na to, budding cells, mga fragmented cells na will be phagocytosed, tapos walang inflammation. Okay, in contrast to your apoptosis, ito yung necrosis natin. So nagsaswell yung cell, naging leaky yung membrane, and then the cells will lyse. Okay, pag nag-lyse yung cell na lumalabas yung mga contents niyan. So some of the contents here will actually incite the mediators in local inflammation, okay? Kaya meron tayong mga inflammatory reaction or signs of inflammation such as redness, ganyan. 'Di ba? Imaginein mo kung may may pigsa or may hubag ka sa buli. O pag may hubag sa buli, init. Kaya ang imo nga blood vessels are engorged, then the blood flow is increased. Mainit yung blood mo, kaya init. Ay, init gali ang ako nga hubag. Yun. Kasakit, may pain, may dolor, may calor, may tumor, swelling kasi lumalaki. So, yun yung example ng necrosis natin. Magkaiba sila sa apoptosis because apoptosis is actually programmed cell death without inflammation. So, ano mga features ng apoptosis natin? Again, loss of mitochondrial function and caspase activation. Remember, itong caspase na to, sila yung mga executioner. Sila yung maggawa ng train of events leading to the execution of the cell. Oh, ito, execute na natin ito. Patalikurin na yan. Okay. Igapos na ang cell na yan. Okay. Sila yon na caspase. And then, yun, pag na-activate niyo yung caspase, eventually this will lead to fragmentation of your DNA and shrinkage of nuclear and cell volumes. Okay? Magiging pycnotic yung nuclei niyan. And then the cell membrane changes. Okay? Magkakaroon ng blebbing. And then eventually formation of apoptotic bodies which are eventually eaten up by your phagocyte, by your macrophages. So, yun yung features ng apoptosis natin. I think yun na lang last slide ko. So, I wish or I hope na may naaral kayo dito. Prepare for the exam which will be on December tuloy. In February 16, that's 5:30. The activity for this topic will be on February 15. Okay, isipin ko pa kung mag-synchronous ako during the next lecture. Pero mahirap pala mag-synchronous, especially sa iba din na hindi din makakonect. So maybe I can pre-record na lang din yung mga subsequent natin. Pag may kailangan talaga na synchronous, okay, yun, mag-synchronous na lang din tayo. Okay guys, good luck on your exam. Make me proud. Ang passing natin dapat yung aim nyo is 100%, okay? So, thank you for listening. So, this is the end of my topic. Thank you.